Hi, everyone. I'm Ian. It's my pleasure to introduce Dr. Ryan Stowers from the Department of Mechanical Engineering. Ryan did his graduate work at UT Austin in biomedical engineering and then went on to do his postdoc at Stanford. Throughout his academic career, Ryan has used novel biomaterials to study 3D cell culture and specifically how biophysical cues from the extracellular environment affect cell fate. In particular, his work has provided novel insight into how physical properties of the cellular microenvironment drive biological processes, such as stem cell differentiation and cancer progression, and a lot in between. Ryan started his lab here at UCSB last fall, where he'll continue to investigate how physical properties of the extracellular matrix drive changes in cell fate during both development and disease. So with that, I'd like to go ahead and welcome Ryan for his presentation today.
All right, thanks, Ian. Can you guys hear me okay? Awesome. So let me pull my slides up, and I just want to say thanks to the organizers of the retreat. It's really an honor to get to speak here. I've met some of you. I recognize some faces, both students and faculty, but as Ian said, I'm relatively new. I started my lab here last fall, so particularly because of COVID, I haven't met many of you, and I'm anxious to chat more about research. So if you find this interesting, we're recruiting students if you're interested in a rotation or if you just want to collaborate. And for faculty, certainly I'd love to hear more about how we could get some collaborations going. My talk today is going to focus on how extracellular matrix viscoelasticity can regulate cell fate. And I'm hoping to get to two quick stories, both from my postdoctoral work at Stanford and areas that we're continuing in my independent lab here at UCSB. So as a quick primer, as many of you are aware, we have cells that exist in the body and in a surrounding extracellular matrix. So this matrix is composed of a number of different biopolymers, and the cell can interact with these, secrete them, degrade them, migrate through them. Everything it does kind of has to interact with or is influenced by the ECM. And so what my lab does is try to design 3D hydrogels to control some of these properties of the extracellular matrix. Things that you may have heard of before, like stiffness or composition, and other things that maybe are kind of newer for the field, things like viscoelasticity or stress relaxation that we'll talk about today. And what's important is that these mechanical and material properties can actually drive differences in how cells behave. So things like what kind of shapes they adopt, whether or not they migrate or divide, and even things as influential as whether or not they differentiate down a certain lineage, or whether they can cause disease progression for things like cancer. And so today I'll tell you a quick story about how we found that matrix stiffness can actually regulate tumorigenesis in a breast cancer model, particularly by regulating the epigenome. And then I'll transition to this concept of stress relaxation and how we can incorporate that into 3D hydrogels and look at how that might affect things like stem cell differentiation. And I should also say, if you have a question, feel free to jump in, or I think I have some time left afterwards, and then there's always the Slack if we don't get to everything. So this first story that I'll tell you about is related to how tumor stiffness can actually drive cancer progression. And this is something that's actually pretty ancient in terms of concept. So this is the Edwin Smith Papyrus. It's the oldest surviving medical text that we have on record. It's over 3,500 years old. It's from ancient Egypt. And along with other kinds of descriptions like magical incantations to cure certain maladies, there's a description of a palpation of a breast tumor. So the physician here understood that if you feel a hard lump in a soft tissue, that there's a potential severe consequence associated with that. And I would argue that this is probably the oldest accurate diagnostic that we have on record. So, people still find many soft tumors or soft tissue tumors with the same methodology. They feel a hard lump in an otherwise soft tissue and then go get it checked out. So we actually do the same kind of diagnostic technique now with a bit more sophistication using things like ultrasound elastography. So here you can scan the breast and see the stiff, dense lump in the soft tissue surrounding it. And you can use that to kind of scan and screen for these tumors. And so the reason that the tumor stiffness becomes so elevated over the course of the disease is that we have extracellular matrix remodeling going on. So here we have normal, pre-malignant, and malignant mammary tumor tissue or mammary tissue in the case of the normal state. So here you can see the extracellular matrix is stained in red. It's very sparse and organized in the normal tissue case, but we get a huge secretion of extracellular matrix when we are looking at a malignant tissue. And not only do we have more extracellular matrix secreted, but we have a realignment or remodeling of those extracellular matrix fibers. So in the normal case, we have these sparse and loosely connected fibers. But in the malignant case, we have these highly aligned fiber bundles. And so both of these two factors, the increased deposition and the remodeling and reorganization of those fibers, result in an increase in stiffness or elastic modulus of over an order of magnitude. So we go from around 100 Pascal to well over a kilopascal. And so for many years, or maybe since we've known about tumors, we've known this fact that they become stiff, but it wasn't apparent until the last few decades that actually this stiffening effect could drive disease progression and not be a mere byproduct of the disease. So a seminal study demonstrating that uses these MCF10A cells, which we heard about in the last talk. These are breast epithelial cells, and they're considered non-malignant. So if you put these cells in a matrigel overlay, they'll develop what's known as a mammary acinus. So these are cells that polarize and form an epithelial sheet, which you can see here. And so this is a three-dimensional construct that kind of looks like a donut rotated along the z-axis. So it's hollow in the middle and has a epithelial sheet around the outside. So what this group did, this is work from Val Weaver's lab. They put these matrigel overlays on top of polyacrylamide gels that they could make soft or stiff or really anywhere in between, and actually modulate the stiffness that the cell can feel. So if you put the cells on a soft polyacrylamide gel around the stiffness of the normal mammary gland I showed you in the previous slide, you get these normal looking mammary acini. And these are kind of the in vitro analog of the mammary gland. But if you put the same exact cells in matrigel again, but on a kilopascal or higher stiffness, these cells start to lose their epithelial character. So they begin to hyperproliferate. They lose that hollow core, and they lose their apical basal polarity, and they start to invade and migrate. So just by changing the matrix properties to look more like what we see in a tumor, we've actually changed the phenotype of these cells so that now they become proliferative and invasive, much like what you see in the tumorigenic state. And so a lot of work's been done on this model and in others regarding mechanobiology and mechanotransduction, demonstrating how interactions with the extracellular matrix shown here can signal through things like integrins and growth factor receptors down these known canonical pathways and ultimately give rise to changes in gene expression, particularly through activation or relocalization of transcription factors. But what wasn't so clear and is just now emerging is that there could be an underlying epigenetic change associated with this as well. And for that reason, or the reason for that is that we have this tight mechanical connection between the extracellular matrix through the integrins and cytoskeletons down to the nuclear lamina, and actually that interacts directly with chromatin itself. So you can imagine that if you have mechanical changes on the outside, those could be propagated through this tight mechanical connection to give rise to changes in chromatin organization, which could have huge implications for gene expression downstream. So that's a question I wanted to explore. To do that, I used this model system developed in my postdoc lab, which relies on alginate and matrigel, forming an interpenetrating network in 3 dimensions. And briefly, the way this works is that we have cells that can bind to the matrigel proteins like laminin and collagen 4. And within that matrigel network, we can actually co-polymerize a network of alginate shown in blue that's cross-linked by calcium ions. So we can control the number or amount of calcium ions that we incorporate into the system and effectively control the stiffness. So we can make very soft gels or stiff gels resembling normal or tumorigenic tissue. And we can do all of that without changing things like ligand density. So it's the same concentration of matrigel or pore size. So on the scale that the cell can feel, we haven't changed anything about the macrostructure of the gel. So again, if we put these MCF10A cells, again, these are non-malignant mammary epithelial cells in a soft or stiff gel, they recapitulate these phenotypes that I previously showed you. So the normal mammary acinus or the invasive proliferative structure in the stiff gel. So throughout the talk, I'll show you these images, which are just a collection of outlines from these structures to give you kind of a feel for the array of morphologies that we typically see. That's what's shown in black and white. Also this metric roundness. So the closer roundness is to one, the more of a perfect circle these structures are. The further it departs from one, the more malignant we say the phenotype is. And then also a invasive metric. So higher percent invasiveness indicates a higher malignant or tumorigenic phenotype. So the first thing I looked at was how the nucleus itself changes between these two states. So I did some high resolution imaging of nuclei and then imported that into Imaris and can actually calculate point by point the local curvatures that exist on the surface of these nuclei. And what we found is that cells in stiff gels have highly wrinkled nuclei. So they have these ridges and caves or cavities that form, and these are largely absent for cells cultured in soft conditions. So you can see at the extreme ends, highly negative curvature events and highly positive ones occur much more prevalently for cells in stiff gels. We can also look at the chromatin organization by TEM. And interestingly, what we found, so this is the outline of the nucleus for a cell in a stiff gel, are these pockets of heterochromatin that pop up at the nuclear lamina that really don't exist at all for cells cultured in soft gels. We didn't find many of these instances at all. Again, we can plot those and see statistically significant differences between those distributions. And so the big question was, it seems like microscopically we can see differences in nuclear shape and also chromatin organization, but what specifically is being reorganized in the genome? So I used an approach called ATAC-seq, which many of you are probably familiar with, but my engineering colleagues are less so. So quickly, the way this works is that we have this enzyme called TN5 that can cleave DNA wherever it's accessible. So in sites where chromatin is open, this enzyme can come in, bind, make its cut, and then we have a sequencing barcode attached. And so we can get a map of where chromatin was opened, and where we have these compacted regions of chromatin, the enzyme doesn't bind readily, and we don't get many sequences. And particularly, one thing to note is that this is amenable to very low numbers of cells, so on the order of 500 to 50,000, which is great for our 3D cell cultures, where we don't have tens of millions of cells like some of the other assays that you may use to determine this type of information. So we can go through there and look at the differences between chromatin accessibility site by site between soft and stiff gels. Each of these orange or tan regions indicate a statistically significant peak. So we see an increase in accessibility for cells in stiff gels at the transcription start site for a number of genes. So these are just 3 instances, but of course we can do this genome-wide. And what we found is that we have over 1,600 differentially accessible regions that are increased in accessibility for cells cultured in stiff gels over soft. And so given that, we see this difference in stiff ECM driving increases in chromatin accessibility, and we wanted to know what's actually binding in this open chromatin site and what are the consequences of that difference in chromatin accessibility. So we took all of those differentially accessible sites and ran a de novo transcription factor motif analysis. So this is basically scanning all those sites and looking for statistically significant motifs that arise at a higher frequency than you would expect. And so what the program spit out is this particular motif as the #1 by a long shot motif. And so if you compare that to existing or known transcription factor motifs, you can see that it looks a lot like this SP1 motif and actually resembles a number of SP-KLF family motifs. But SP1 was the most likely target. And so SP1 is particularly interesting because it's a prominent transcription factor in a number of pathways, but most notably for us is that its role is pretty well established in chromatin remodeling. And also, if you look at what diseases are associated with SP1 gene targets using this database, the #1 disease that pops up is malignant neoplasm of the breast. So this is early malignant transition of the breast, which is kind of the model of tissue transition that we're looking at with this 3D culture model. So we first verified that we're actually seeing an increase in binding or presumed binding of SP1 to our open sites by doing transcription factor footprinting. So basically just scanning these regions around these SP1 motifs and looking for a disruption in accessibility, which you can see centered on the motif here for cells in stiff gels. We further validated that by looking at or showing a 2 to 3-fold increase in SP1 phosphorylation for cells in stiff gels. And also that SP1 gene targets are increasingly upregulated for cells in stiff gels. And these are particularly targets associated with this malignant neoplasm of the breast that I just mentioned in the previous slide. Sorry, was there a question? Okay, so we have some indirect evidence showing that we have some motif enrichment, footprinting, increased phosphorylation, increases in target gene expression. So all of these are kind of funneling down to SP1 having a strong role in promoting this malignancy. But to determine that directly, we knocked down SP1, put those cells in a stiff gel, and we saw that the phenotype was reverted. So they're no longer highly invasive or proliferative, but in fact, they're much more rounded and less invasive. Just to confirm this, we looked at another cell line. So these are, again, normal human mammary epithelial cells. If we inhibit SP1, that's with this mithramycin A compound, put those cells in a stiff gel, these cells are no longer highly invasive or proliferative. And interestingly, using 2 cancer cell lines now, we again see an effect, diminishing proliferation and invasion of these cells when we culture them in a stiff gel with mithramycin, the SP1 inhibitor, compared to the control. So this is with MCF7 and MDA MB231s. And so getting back to this epigenetic component, we looked at what proteins actually interact with SP1. So this is a top 10 protein-protein interaction database from a string analysis. And what's particularly notable is that 4 of these are chromatin modifiers, histone deacetylases in particular. So I'm not going to show you the data, but we showed an inhibition of HDAC class 1 reverts the phenotype back, just like what we saw with the SP1 knockdown. And if we further go and knock down each of the individual members of that class, 1, 2, 3, and 8, we can see an effect on phenotype if we knock down HDACs 3 and 8. Again, we see this rounded phenotype that's not particularly invasive. And notably, if we knock down HDACs 1 and 2, which are actually the more well-studied HDACs in this family, we interestingly didn't see nearly the same extent of knockdown, of phenotypic reversion. So the cells remain invasive and highly proliferative. And so we can kind of arrive at this simple model where we haven't filled in all the details, but we have some more insight where we have some stiff extracellular matrix signaling through integrins and PI3K, ultimately giving rise to differences in HDAC associated, particularly HDAC 3 and 8, chromatin remodeling, and SP1 binding to those open chromatin sites. All of this driving this increase in invasion or tumorigenic phenotype. Okay, so I'm going to switch gears a bit from that story, which was focused mostly on matrix stiffness, and focus on another mechanical property that's particularly interesting for our group, and that's this concept of stress relaxation. So stress relaxation, you can think of it like the behavior of silly putty compared to a rubber band. So when you stretch a rubber band, it stores energy much like a spring, and we call this concept elasticity, or we would describe that material as highly elastic. When you stretch silly putty, on the other hand, it dissipates energy over time in a viscous manner, and so we would describe that as a viscous or a viscoelastic material. So it's going to dissipate energy over time. And why this matters is that most soft tissues in our body actually are highly viscoelastic. So here we're plotting adipose, liver, brain. You can see that if we apply a constant strain to this material, so we shear it to 10 or 15%, and then we look at the stress over time, we see that that stress drops pretty dramatically. So over the course of 10 to 1,000 seconds, we get a almost full decay in the stress, much like what we would have when we stretch the silly putty and then look at the energy that's stored in that system. And this is particularly notable because many of the hydrogels that we use to study these systems, in my field in particular, are covalently cross-linked. So they behave like the rubber band and not like silly putty. So my postdoc advisor actually developed a series of gels that we can encapsulate cells in 3D and vary the stress relaxation time. So this T-half is a metric that refers to the time it takes to reach 50% of the original stress value. So if you look at these, you can see that collagen relaxes stress, let's say on the order of 10 seconds. Liver, adipose tissue are on the order of 100 seconds. And this covalently cross-linked gel, goes to infinity. So if we can vary the time between, let's say, around 100 seconds to over 1,000 seconds, we can kind of encompass this range that we see between soft tissues and covalently cross-linked hydrogels. And what's interesting is that if you put mesenchymal stem cells or mesenchymal stromal cells in these 3D matrices, you see a difference in differentiation based on this stress relaxation. So we have the cells in a relatively stiff gel that's known to promote some degree of osteogenesis. We can stain that for alkaline phosphatase, which is an early marker of osteogenesis, and see some degree of staining. But that staining or differentiation is significantly enhanced as we increase the rate of stress relaxation or make these gels more viscoelastic. So in my postdoc, I looked at this, the mechanism underlying this with a lab mate, a grad student, Hongpyo Lee. And what we noticed about cells in these gels is they seem to expand their volume or just become quite large when we allowed them to grow in stress relaxing matrices. So if we look at the cells at day 1, they're roughly spherical after encapsulation. In slow relaxing gels, so these are the more elastic types, these cells retain that morphology, so they stay spherical. But as we increase the stress relaxation rate, you can see that the cell volume expands pretty dramatically. And so if we quantify that, we can see that we get a 2 to 3-fold increase in the volume of single cells over the course of a week when we allow them to grow in these relaxing matrices. And so the reason for that is that in an elastic matrix, the cell's kind of confined and trapped. It's almost frozen because it can't remodel its environment. But in the stress relaxing matrix, it can physically deform its environment, and that allows it more room to grow and spread out and increase its volume. So we showed that if we artificially confine the cell, even though it's in a fast relaxing gel, we can add a high osmotic culture media and restrict the cell from growing. We can actually inhibit its ability to expand its volume. And what was notable was that we saw a decrease in this differentiation capacity as we did that. So instead of having 70, 80% of cells undergoing osteogenic differentiation after a week in the fast relaxing gel, as we dose dependent decrease its ability to expand its volume, we see less and less differentiation over time. We identified a calcium channel that actually regulates this process called TRPV4. So we saw a decrease in TRPV4 expression as we decrease the rate of stress relaxation. And functionally, we saw a decrease in the level of calcium that's imported into the cell as we decrease stress relaxation rate. So when the cell is able to expand its volume, it's activating this calcium ion channel, bringing in calcium and also water associated with that, expanding its cytoplasmic volume and undergoing differentiation. So I'll quickly get through the last few slides here. If we inhibit that channel, TRPV4, even though the cells are in a fast relaxing gel, we can effectively abrogate differentiation. Similarly, if we put the cell in a confining matrix and then activate this channel with a small molecule, and we can drive differentiation and some degree of volume expansion, even though the cells are otherwise entrapped. And ultimately, how this occurs, many people have postulated that YAP is driving many of the mechanotransduction pathways or differentiation events. And so we looked at whether or not YAP is going to the nucleus to a different rate between fast relaxing gels. I think that says 5 minutes. Okay, we're good. In fact, we didn't see a increase or any change at all in YAP translocation to the nucleus between slow and fast relaxing gels, or when we inhibit volume expansion by this osmotic pressure. But we did see a significant increase in nuclear localization of RUNX2, which is a potent master regulator of osteogenesis. So when cells undergo volume expansion in fast relaxing gels, we see a massive increase in RUNX2 in the nucleus. When we confine cells by osmotic pressure and prevent them from expanding their volume, even though they're in these fast relaxing gels, we see exit of RUNX2 from the nucleus, and it's mostly localized in the cytoplasm. And so you can see those trends plotted here, where we have almost no trend with YAP compared to cell volume, but a pretty substantial correlation between RUNX2 in the nucleus and cell volume. So we wind up with this model where we have a cell in a three-dimensional environment, pushing, pulling on its matrix through integrins and other receptors in a viscoelastic gel that allows for physical deformation or physical remodeling of the environment, cell spreading and volume expansion that's mediated through this TRPV4 channel that I told you about. And that ultimately allows RUNX2 to go into the nucleus and upregulate osteogenic genes. If we inhibit any portion of that pathway, so we inhibit TRPV4 signaling, or we inhibit volume expansion by using elastic matrices or this artificial osmotic pressure, we can effectively abrogate all of that differentiation. So I just wanted to draw your attention to a recent review from my postdoc advisor, Ovi Chowdhury, and others that just came out in Nature a couple weeks ago. So this highlights the effect of ECM viscoelasticity. So that's the stress relaxing effect that I told you about on cell behavior. For this biology crowd, I think this may be a new concept for some of you, but this review does a really great job of highlighting the importance of these things and how they're distinct from things like stiffness or modulus of elasticity that maybe is more familiar. So I'll just wrap up by acknowledging first my nascent and growing group here at UCSB. So Ian is who introduced me, MCDB student, second year, who joined my lab through the rotation program. And Philip is a ME student. So if you're interested in doing a rotation, certainly send me an email or otherwise talk to me. We're always looking for new students. And then, as I said, most of this work came from my time at Stanford. So in the lab of Ovi Chowdhury, the last story I just told you about was a collaboration with Hong Pyo Lee. And the first epigenetic story was basically everyone else listed here contributing to that. Funding for that was through NCI Fellowship and R01, and some other funding for that from the ACS and an internal grant at Stanford. So with that, I think we have some time for questions. I'm happy to hear them. Thanks.
Yeah, there are a few questions that were put in the Zoom chat. The first one I see is, does integrin density on cell surfaces alter the degree of stiffness required for the response?
That's a good question. So we didn't check the degree of integrin density or clustering that's associated with that, but certainly a number of other folks have looked at the effects of integrin clustering and how that affects downstream signaling. And I would be surprised if it wasn't an important factor. But it tends to associate with things that give rise to mechanotransduction. So higher matrix stiffnesses generally allow for more integrin clustering. More matrix remodeling or stress relaxation are generally driven by high amounts of integrin clustering. So I think all of these things are related.
Okay, the next question is, does stress relaxation rate affect cell division rate in general?
Yeah, actually, so this wasn't my work, but from the same lab that I did my postdoc in, this student, if you can still see my slides, Sungmin Nam, he had a great paper out in Science Advances looking at the pathway that's involved in signaling between stress relaxing matrices and controlling cell division. So if you're interested, I'm not sure who asked the question, but I could link you to the paper. But he's the first author if you want to check that out.
And then we have one more question that says, do you expect to see similar epigenetic regulation from stiffness variation in other cancer types, e.g. glioblastomas?
Yeah, it's a great question. I don't know for sure, but I would be super surprised if we identified the one model system in which we do see an epigenetic effect linked to stiffness, and it wouldn't be implicated in, I mean, many of these other soft tissue tumors are also driven by things like matrix stiffness, matrix density, these sorts of things. And I would imagine that it's a pathway that's probably conserved across a lot of these different diseases. maybe some of the features change, maybe some of the mechanisms by which they occur change, but I think it's probably a ripe area to explore and something my lab's definitely looking into going forward.
Okay, there's another question. It says, I'm not exactly sure how to pronounce this, so I apologize in advance if I don't say this word correctly, but have you studied the lamina plastica associated with gastric cancer as a process of extracellular matrix modification?
Yeah, I don't recognize that term either. So no, I haven't studied it. I'm not sure who asked the question, but I'd be happy to chat about it. I really don't know anything about it though.
I have a question I'm having a hard time phrasing via typing, so maybe it's a bad question. I'm wondering, the SP1 sites, you see more of them uncovered. Do you think that's some sort of active targeting or active uncovering of the SP1 sites, or do you think it just so happens that coincidentally those are the ones that are uncovered and thus that's what gets activated?
Yeah, so I don't have any direct evidence, but I would imagine that it's probably an active process. I think it would be pretty coincidental that we like randomly stumbled upon that using the pipeline that we used. I mean, we weren't selecting for single cells or only selecting for invasive or highly proliferative cells. We just looked at all the cells in the gels. So I imagine there's some active process driving accessibility through SP1. So one way that could happen is that SP1 is known as a pioneering factor. It can actually open chromatin itself in some cases. So the simplest model would be that it drives chromatin, it drives an increase in accessibility in chromatin sites where it's going to bind. But it could be facilitated by binding partners. SP1 is also known to interact with a number of chromatin modifiers. So which way that goes, I'm not really sure, but it's something we're pursuing.
Awesome. So the model is that it's kind of both upstream and downstream of the chromatin modification.
Well, so if it's acting in this pioneering fashion, then it could be opening chromatin itself, binding there, and then driving expression. And so that's the simplest model. If it's not that, then yeah, it is a bit puzzling how it's working on both ends. But so I don't have any evidence to say one way or the other, though. But it's definitely a great question.